All right, are there any questions? So the test is on Friday, okay? And yeah, you have a question or no? Yeah, go ahead.
So I wonder how much detail that is going to be. For example, because I saw a lot of like technical terms. So
you need to know those, yeah. Technical terms you need to know, yeah. You need to know. I mean, like, yeah. I thought you were going to ask me about the detailed structure of the DNA bases. You know, that probably you don't need to know. Like AGCT, yeah. I'm not, but yeah. Like what technical terms?
Like the gel electrolysis.
What?
I mean,
Gel electrophoresis. Yeah, yeah. You need to know that term. Yeah. Yeah, gel.
Just want to double check it's everything before transcription and translation.
Yeah, it ends with translation, basically, yeah. Everything before that, yeah. So this is your opportunity to learn that stuff, you know, and again, you know, yeah, it is challenging because in 6 weeks we are covering all of molecular biology, right? But, you know, see, you're already trained as an engineer, right? Right, and you're almost close to your PhD or whatever. So nobody's going to, you're not willing to spend another, you know, whatever, 6, 7 years learning all this stuff, right? I mean, so we have to go at a much faster pace, right? But it's not that bad. I think it's structured. People before you have survived, so I think you can do it. You know, so just go ahead today and tomorrow. Just study like crazy, right? So and yeah, forget about everything else. Right? Just focus on this, because I mean, especially for people who are working at the going to be working at the interface of biology and engineering, right? You all of you are good engineering students. Otherwise, you wouldn't come here. Okay, there are easier courses to take. All right. So you're good in engineering, but your biggest handicap will be the lack of knowledge of biology, right? Even what I'm covering here is not enough, right? The specific problem that you will look at, you'll have to learn more about that. That's the domain knowledge, right? But this is the bare minimum, right? And you are in better shape because I had to learn these things by myself, okay? I mean, I had off from teaching for a couple of years, but I had to learn and then publish papers within the 2 years, right? That was the requirement, right? And, you know, I attended a class where it was an honors section of Biology 213, and the professor came and said he wanted to teach us how to do research. You know, he wasn't going to cover the routine material, right? Because it's an honors section, 213 honors section, right?
So you were sitting as a student?
I was sitting with all the undergrads, okay? Not at your level, okay? Much lower, okay? This is a 200 level course, right? Then I took another one, I think it was Genetics 310, okay? So 200, 300, and then I took another course. I was sitting in on another course that was Nutrition, I think 470 or something like that, okay? So I've been through that, okay? And that is after, you know, this was in 2001, okay? So I had already been a faculty, I had been teaching for 10 years, right? I had been teaching already for 10 years. So if I could do it, you know, at that time, like I was like 37 or something, so you can do it. You guys are much younger. You can work harder, okay? You can sleep less and work harder and do it, you know? No, and because you're trying to do something that most people cannot do, right? So it is going to, the price is going to be higher, right? Because, you know, if you learn this biology, right, then you'll be in better shape than most people can talk about it. You know, everybody knows, oh, doing interdisciplinary work is important, but to actually get the work done, you will have to get into the gutters and be able to, you know, make the connections and all that. And for that, you need that knowledge, right? So, I mean, I have to admit, you know, during those 2 years, I learned the bare minimum. And when you're talking about remembering terms and all that, because I had other things to do also. So the way I really learned it is by lecturing in front of people like you, okay? Once that thing got over, I offered a course in the summer of 2004, okay, which is the forerunner of this course, right? But then when you're lecturing, then you have to learn, okay? You're forced to learn, okay? Right? And, you know, you'll build on that. If you're working on some cancer, you need to know details about that. You don't want to be thrown off when somebody talks about pathways, talks about, you know, ligand substrate interactions, or gel electrophoresis, or next generation sequencing. Some of you have, I mean, many of you are associated with the new center, right? We do have the state-of-the-art next generation sequencing facilities over there, okay? And it's exactly the same kind of sequencing that I covered here, except that you first cut it down into pieces, right? And then the same kind of technique is used basically to figure out the complementary sequence, right? So if you, I mean, I don't know whether it made sense to you or not when you took a tour of the center a few weeks back, right? But if you didn't, just go and take a look. There is a chart in the coffee room area, right, that talks about the different steps. So right now you will be in a better position to go and take a look at that chart and understand what's going on, right? Okay, but there is no way out. You have to learn this stuff. Okay, I can lecture, I can double the lecture time, I can keep on talking. It's not going to help, right? You have to pick up the terms. That is the choice you made, right? So, all right, but it's a one-time thing. It's not like you have to keep on learning. But if you learn it and deliver on the exam, then that way, this is a coupled reaction. Okay, you've coupled your learning process with getting a good grade. Okay. Right? I mean, normally the reaction will not take place. Okay, you are outside your comfort zone. Okay, the free energy is positive. Okay, to go and learn all this stuff. So couple the two together, and put your knowledge in this course to work for you. Right? Okay, so any other questions? Now today after the class, like during my office hours, all right, I probably will not be available for some of the time because I have to meet somebody. Okay, but if you need to see me, then just send me an email. Right? I will probably be on campus tomorrow, so if you need to see me about anything, but I need to see somebody between 3 and 3:30 this afternoon, so. Now, towards the end of the last class, all right, we were discussing how PCR can be used in forensic science to track the perpetrator of a crime, right? Now, the DNA sequences which create the variability that is exploited in this type of analysis, it contains runs of short repeated sequences such as GT, 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 right? And the number of these repeats, it is highly variable in the population. It varies from 4 to 40. Like you might have 5, I might have 25, okay? And at different loci, it's going to be different, right? There are several places where you have these repeated sequences, right? Now, a run of repeated nucleotides of this type is commonly referred to as a VNTR sequence, where VNTR stands for variable number of tandem repeats. Now, because of the variability in these sequences, each individual will usually inherit a different variant of each VNTR locus from their mother and from their father. And therefore, two unrelated individuals will therefore not usually contain the same pair of sequences. Okay, but if you're related, like your brother, sister, or your parent or something, there probably will be some common stuff. So that's why they could use DNA from a relative to track somebody down too. Right? But it won't be a perfect match. So how is this technique carried out? A PCR reaction is run using primers which bracket the VNTR locus, and that produces a pair of bands of amplified DNA from each individual. One band will represent the maternal variant of that locus, the other one the paternal variant. And the length of the amplified DNA is going to depend on the number of repeats. Okay, it's between 4 and 40. So if you do gel electrophoresis, all right, the position that it will migrate to depends on the length of the DNA sequence. Right? So this fact can be used to narrow down the list of suspects in a criminal investigation. Right? And this is how it goes. The steps are as follows. Yeah, go ahead. Yeah. Yeah, the primer will bracket only, you will only bracket the repeated part, yeah. Because you don't want, see the entire genome is like 3 billion nucleotides, so you don't want to go and run PCR on the entire genome. It's going to take too long, it's expensive, it's a waste of time, right? But the places where the variability is going to happen, you bracket that area and you make many, many copies of that DNA, right? Amplify it so that you can run gel electrophoresis, right? So the steps are as follows. First, select a number of VNTR loci, right? Then use primers to bracket each locus. Don't use just one locus, right? That's not good enough. You need a number of them, right? Like you want to have some more information so that you make a good decision, right? Now amplify the DNA using PCR, and then you run gel electrophoresis. So this process should be carried out on DNA obtained from the forensic sample and also DNA samples from each of the suspected individuals. If the bands obtained during gel electrophoresis on the forensic sample do not match the bands obtained from a suspect, then this particular suspect can be eliminated from the list of suspects, right? And this is how, like, you hear about somebody that spent 20 years in jail, right? And now they've been freed based on this DNA technology. They see the bands don't match, and that technology probably wasn't there when these guys ended up in jail, okay? So you can, but of course, if there is a match, there is still a possibility that it could have come from somebody else, right? So that's what will give the lawyers, a lot of room to argue. And with some good mathematicians or statisticians, they'll say this is the probability and all that, and there could be somebody else that did it. Okay, and the job of those lawyers is basically the defense lawyer is to confuse the jury, right? In fact, in this context, I should probably mention that Like I have served on a jury twice. Okay, so you know, like this was a you know, driving on. Let's see, driving while intoxicated case. Okay, there is a difference between the two. Okay, between driving while intoxicated and being drunk. Okay, intoxicated means you have lost your mental faculties. Okay, and there is a blood alcohol level, right? I think it's 0.08 in the state of Texas. Okay, if you're above that, then you're legally drunk. Now the problem is that when they measure your blood alcohol level, okay, in the hospital, and this was an accident where the person was really badly injured, had lost a lot of blood and all that. So when they measure your blood alcohol level, okay, they measure the serum alcohol level. Okay, that means they take out the watery part, and so the number is different. So you cannot apply the 0.08 test to that. Okay, and they would not give us the correspondence, so we didn't know. Okay, and we cannot go and look on the internet and find out, because then we'll get into trouble, right? Because, you know, the defense lawyer and the prosecution, they have to agree, right? So they agreed that, okay, you know, we'll leave this ambiguity, let these guys make a decision. Okay, so we had to make a decision based on the numbers they gave us, but we don't know what is the conversion factor. Okay, how do you convert the serum alcohol level to the blood alcohol level? Okay, but ultimately we use other evidence to come to a decision. But I'm just saying that, so, you know, people, and then they put another guy in front. Okay, that guy's a lab technician, right? He's the one that did he took the measurements and all that, you know. But then the defense lawyer is asking him stuff like, you know, to convert this from one to the other, and he cannot, right? But I mean, we were all educated people sitting over there, so we could figure this out. But I'm just saying that those are the kind of techniques that people use, you know, that they're trying to question his credibility, because that guy probably just takes the readings, he looks at some chart and does it, okay? But, you know, it's like converting from milligrams to centigrams or something like that, you know. So it was really quite weird, right, to see how things can go. You know, so. And you know, that was for a, you know, driving while intoxicated case. Okay, so if the lawyer is doing that in that kind of a case where the penalty is not that severe, okay, you can imagine what they will do in a murder case, right, to try to, you know, get their client free, right? So they do all kinds of stuff. All right, any questions? So now we move on to genetic engineering, where you're basically trying to, you know, alter, you're trying to create organisms with altered genomes. Right? So the advances that have been made in DNA technology over the last 3 decades have opened up vast avenues for genetic manipulation. For example, using DNA technology, completely novel DNA molecules can be constructed. Right? So to do so, one can cut a plasmid vector using a restriction nuclease. So again, none of this is foreign terminology to you. You know what a restriction nuclease is, all right? You know what a plasmid is, all right? And then you insert a DNA fragment into the plasmid vector using DNA ligase, right? Ligase again is the enzyme that is used to stitch DNA together, right? Now this procedure could be repeated to insert additional DNA fragments onto the plasmid. Another application of DNA technology is the production of large quantities of rare cellular proteins using cloned DNA. For example, if you want to produce large amount of insulin for let's say diabetics, right, to take insulin shots, you know, before the advent of DNA technology, you'd probably have to put it in some animal or something like that, you know, to produce this. Okay, but with DNA technology, you're going to be able to produce these rare cellular proteins using cloned DNA. Right. So to do so, one can make use of a plasmid which already has a promoter sequence inserted into it, right? Such a plasmid is called a double-stranded expression vector. So then after the promoter region, you'll put the sequence for the protein that you're interested in. So the protein coding sequence is inserted into the region immediately following the promoter by using a restriction nuclease. Again, the restriction nuclease is going to cut DNA at certain specific locations, okay, in a very, very predictable manner. And then you use DNA ligase to stitch it up, right? Now you introduce this recombinant plasmid, right, into bacterial cells. Every time the cell divides, the plasmid will also be replicated. Okay. On the other hand, if there is transcription, right, then these cells will be overexpressing the messenger RNA and the corresponding protein, right, corresponding to this thing that you have inserted. Yet another application of DNA technology is the artificial production of RNAs by in vitro transcription. RNA is a very unstable molecule, right, because you know, on our skin also we have enzymes that will degrade RNA, right? So you touch RNA, RNA is gone. Okay? It's not like DNA, right? So most RNAs found in a cell are present only in small quantities, and they are hard to isolate from the other cellular components. So once the gene that is coding for an RNA has been isolated, you can produce that RNA artificially in a test tube by letting RNA polymerase just operate on that gene, right? So you don't have to go inside the live cell. Now the function of a gene is best revealed by an organism which has had that gene mutated in some way. Neither the complete nucleotide sequence of a gene nor the three-dimensional structure of a protein is sufficient to deduce a protein's function. There are many proteins, right, such as those that have a structural role in the cell or which normally form part of a large multi-enzyme complex. You will not be able to see any activity by itself, right? So you don't know what the function of that protein is. However, if you create a mutant where that protein is not there, then you will very easily see what's happening, right? For example, if you knock out the gene for insulin, you will see a diabetic mouse, right? So I mean, of course, in the case of insulin, the function was known, but in other cases, it may not be that obvious. So mutants that lack a particular protein may clearly reveal what the normal function or functions of that protein are. Prior to the advent of recombinant DNA technology, these mutations would have had to arise naturally, and so would necessarily arise by chance and be unpredictable. Like if you're studying a disease, okay, somebody would have to have that disease, right? Somebody accidentally got that mutation, has that disease, then only you can study it, okay? You cannot, simulate that disease in the lab, okay? But today it's possible to do that. So recombinant DNA technology makes it possible to introduce precise mutations by the technique of site-directed mutagenesis, right? So at a particular site, you want to change a DNA base, right? And create mutant DNA, you can do that, right? Today with recombinant DNA technology, right? And this is how it is done, okay? See, like, and again, I mean, there is a textual description that will follow, but see, this is a plasmid cloning vector, right? So this is the normal gene that you have inserted. In the region of interest, let's say it has got CTG, right? And the complementary sequence is GAC, okay? So this is a double-stranded DNA sequence that has been attached onto a plasmid, right? Now, you separate out the two strands, okay, by heat or by whatever, you separate them out. Then you want to purposely introduce a mutation, right? So what you can do is, this bottom one is a GAC, right? You purposely put in GCC, okay? Put in that. So this is a site-directed mutagenesis, okay? You're starting a mutation. Now, you let the, like DNA polymerase run, okay? So it's going to look at this strand and make the complementary copy. Only there is a mismatch in one location, right? Now, so now you introduce this guy, right? This plasmid you introduce into cells, let's say bacteria, right? Every time the cell divides, this is also going to get replicated, okay? Now DNA replication is semi-conservative. So each strand will serve as a template, okay? So half of these guys will be producing the correct one, right? Complete correct strand. The other half is going to produce a mutant strand, okay? Now if you do transcription and translation, this one which is the correct sequence, that will produce the correct messenger RNA leading to the correct protein. GSC, right? I mean, you can look in the book. It codes for asparagine, right? That is the amino acid, whereas GCC codes for alanine, right? So it's going to be a different amino acid, right? So so half of the progeny are going to be producing the normal protein. The other half is going to produce a protein with a single desired amino acid change, right? So this is the technique that we can do. Now, prior to the advent of DNA technology, the mutations would have to arise by chance. Okay, if you got lucky or unlucky, then you got the mutation, then only you can study. But now you can go and do the exact mutation that you want, right? And then study its properties, right? Using this this kind of technique. And in fact, kits are commercially available for doing this kind of stuff. Right? So in recombinant DNA technology, one can start with a cloned gene and proceed to make mutations in it in vitro. That means in the test tube. Okay, you can do that. Then by reintroducing the altered gene back into the organism from which it originally came, one can produce a mutant organism in which the gene's function may be revealed, right? So in the previous slide, the gene to be mutated is first inserted into a plasmid, and a sequence GAC, say in one of the DNA strands in the gene, is replaced by a mutated sequence GCC. I showed that to you in the picture. The plasmid is then introduced into a cell. Due to the semi-conservative nature of DNA replication, every time the cell replicates, one half of the progeny will end up inheriting the normal gene, while the other half will end up inheriting the mutated gene, right? And the result is that half of the progeny will produce the corresponding normal protein, while the other half will produce the mutated protein, right? So you have been able to engineer this mutation, right? Very, very precisely, right? Now, recombinant DNA technology can be used to carry out gene replacement. You could replace a gene entirely, okay? You could do a gene knockout. That means you could just take the gene out altogether, right? Remove its function completely, or you could add a gene. You could put an extra copy of some gene, right? Now, organisms into which a new gene has been introduced, or those whose genomes have been altered in other ways using recombinant DNA techniques, they are known as transgenic organisms, right? Now, it is clear that for haploid organisms are organisms that have only one copy of each gene, right? So, it is clear that for haploid organisms such as bacteria or yeast, transgenic progeny can be produced quite easily, okay? All you have to do is just basically engineer that one mutation, right? And you have it, because even at the phenotypic level, because it has only one copy of each gene, you will see the result of disrupting that particular gene, right? Or messing around with that, okay? So for diploid organisms such as mice, right, or even humans are diploid, you have two copies of each gene, right? So it is more difficult but still possible to achieve gene replacement. So if you want to create like a diabetic mouse, right, what you have to go and knock out both the genes that code for insulin, okay, in each and every cell of the body, right? So if it's going to be in each and every cell of the body, right, it's not enough to, take an adult organism and try to, it'll have to be at the egg and the sperm stage, okay? So you'll have to go through several cycles, right, to do that. So this involves the following 4 steps. First, an altered version of the gene, basically with the mutation that you're interested in, is introduced into cultured embryonic stem cells, right? A few rare embryonic stem cells will have their corresponding normal genes replaced by the altered gene through homologous recombination. Not all of them, some of them, okay? Although often laborious, these rare cells can be identified and they can be cultured to produce many descendants, each of which carries an altered gene in place of one of its 2 normal corresponding genes. But only one altered gene, right, in these embryonic stem cells. But now you have to get it back into the organism, right? You have to get it back into the organism, then get it to the egg and the sperm, and then ultimately get a full-blown organism that will have the gene altered in each and every cell of its body. So these altered embryonic stem cells are then injected into a very early mouse embryo, right? As a result, the cells are incorporated into the growing embryo, and a mouse produced by such an embryo will contain some somatic cells. Somatic cells are cells of the body, that is cells other than the reproductive ones that carry the altered gene, right? So some of these mice will also contain germline precursors of reproductive cells that contain the altered gene. Because remember, you have to, so having some cells, that gene altered in some cells of the body is not good enough. Okay, for that organism, in its egg or sperm, there has to be an altered gene. Okay, you have to get it to that. Okay, so some of these mice, all right, will contain germline precursors of reproductive cells that contain the altered gene. Now, if you get such a mouse to breed with a normal mouse, some of the progeny will contain the altered gene in all of their cells, because when the egg and the sperm fuse, all right, then one gene comes from the father, one gene from the mother, okay? So if the egg has like a mutated gene, right, then the fertilized egg is going to have that one mutated gene. So the organism that develops is going to have that one mutated gene in each and every cell in the body, okay? But still not 2. Yeah, go ahead.
When you say gene, you're talking about modifying just one chromosome, the gene in one chromosome.
Yeah. Gene or maybe one DNA. It could be just one DNA mutation that you're trying to do. Okay. If you're dealing with sickle cell anemia, it's just one DNA base that you're trying to change.
Just in one chromosome.
Yeah. Because the gene will not be distributed across different. Usually not. Okay. It'll be in one chromosome. Yes. Yeah. You mean one out of the 2 chromosomes? Yeah. When I'm talking about one gene, means it's in a pair only one of them has been mutated. Yeah. So when such an organism is bred with a normal mouse, some of the progeny of these mice will contain the altered gene in all of their cells. Okay, so you have moved it from one part of the body, right, basically to an organism that has got one gene altered in all its cells. Okay, now, yeah, this is because 2 haploid reproductive cells of different kinds, the egg and the sperm, have to fuse together to produce the diploid cell, which will undergo repeated cell division to produce the entire organism. Right. So now you have an organism that has got a mutated copy of the gene, but only one copy in each and every cell, okay? Now, if you breed that organism, right, with another one that also has a mutated copy, right, in each and every cell, then there is a 1 in 4 chance, right, that the new progeny will have both copies defective, right? So if 2 such mice are in turn bred, some of the progeny will contain 2 altered genes, 1 on each chromosome, and that answers your question, right, in all of their cells. And this again is a consequence of the meiotic process by which the haploid gametes are produced from their diploid precursors, and the fact that when the egg with an altered gene fuses with a sperm with the same gene altered, then the resulting diploid cell will have both copies of the gene altered. Yeah.
Why don't you do this from the beginning? Modify the 2 chromosomes and that's it. But why do you wait for them to like, reproduce and...
No, because you want to get it in every cell of the body. The only way it will happen in every cell of the body, okay? Like we humans, we have 100 trillion cells, right? If you want to go and make changes, you'll have to do it on all the 100 trillion cells, right?
No,
so
if you do it like when the mice is still in an embryonic phase, you have much more. Well, I mean, the same way you do it for 1 chromosome, you could do it for 2 chromosomes. No?
Probably not, you know. Probably not, yeah. Maybe there are difficulties associated with that, you know. But yeah, to get it in every cell of the body, you basically have to do it at the egg or sperm stage, okay? So that's what the main challenge is over here, okay? And again, remember, these things are not 100% guaranteed, okay? It's saying some of them will produce this, okay? The cloning and all that is not that easy, right? Because most of the time it will not work, right? Because whatever happens in nature, somehow the conditioning and all that, everything is right. Okay, so things take place the way they are supposed to. When we try to do it in the lab, it may not always happen, right? So if the original gene alteration completely inactivates the function of the gene, then we will have what are called knockout mice. Say, for example, if you're looking at the gene that codes for insulin, you go and knock it out in every cell of that mouse, right? You're going to have diabetic mice, right? And there are people that, you know, and these are commercially available, these diabetic mice. I know a guy in the animal science department, right? He works with the diabetic mice because he wants to show that, you know, there are some amino acids like asparagine, right? That they can help. See, if somebody has diabetes, then the wounds don't heal that readily, okay? I mean, the cut stays and it has a tendency to get infected. So he wants to show the beneficial effect of, you know, supplementing the diet with asparagine or something like that, right? To reduce the time that it will take for the wound to heal, okay? So for that he needs diabetic mice, right? So he buys those diabetic mice and put cuts on them and give them this diet and then, you know, figure out how long it takes for the wound to heal, right? So there are people that do those kinds of experiments, even on this campus, right? So these are called knockout mice, okay? If you knock out the gene, the beta globin gene, right? Then you'll have mice. I don't know whether anybody does that or not, but you'll probably have mice with sickle cell anemia, right? But it won't survive. And then there are other people. Those are not knockout mice, but there are people on this campus who study colon cancer, right? So, you know, and they'll buy like 500 rats or something like that, you know, and then give them injections to produce cancers, okay, in the colon, all right? And then put them on different diets and measure the size of the tumors and things like that to see the beneficial effect of diets, you know. All right, any questions? All right, if there are no questions, then we are going to move on to the next chapter, right, which is about cell division, right? And again, you know, whatever I'm covering here, that is just the bare bones, okay? If you want to know more, there are books that are available. In fact, the reference book that I gave you, right? Or you can even Google on the internet and find out more information, you know. So especially in this day and age, there is no, you know, dearth of access to information. Right? So we are going to move on to the next chapter, which is about cell division, right? And because, you know, an important component of this course is the use of signal processing techniques, right, in the context of cancer, right? And that's also an important focus of the center, right? Half of that, I mean, the part of the focus of the center is on plant genomics, okay, to, you know, increase the yield, to, you know, go ahead and make the plants more, use water more efficiently and so on. Okay, and in case you didn't know, okay, there's a guy by the name of Norman Borlaug, right, who used to be a professor here at A&M, right? He's the one who's the father behind of the Green Revolution, right? And I don't know whether Charlie mentioned that before or not. Okay, apparently he has saved more lives, okay, in the history of mankind than anybody else. Okay, because he saved billions of lives because by generating the ability to produce more food, right? And the way he did that is he did some genetic variation whereby he figured out a way to make the wheat plant shorter. Okay, so if it is what was happening before was that it was becoming too tall. If you fertilize, it fell over, and the yield was suffering. Right? So he made it shorter. So that you can fertilize, right, and increase the yield per unit. Okay, considerably. He actually got the Nobel Peace Prize, and he used to be a distinguished professor here at Texas A&M. I think he passed away 2 years ago. Right? Probably at the age of 95. So there is a center named after him. For those of you that will be working on agri-life projects, it's the Norman Borlaug Center is right there. So this DNA technology and all, it has its uses. Okay, but and you can and another major application is in cancer. Okay, so that's why right now we are switching to cell division. We want to talk about cancer, how the process of cell division gets messed up in cancer. So first, before that, we have to understand cell division. So in this chapter, we discuss cell division, which plays a crucial role in the propagation of all life. The only way to make more cells is by division of those that already exist. All right. So there used to be a theory that was called the spontaneous generation theory, right, which said that basically life just arose out of nowhere. Okay. And that theory was put to rest by Louis Pasteur by an experiment. Right? See, because people tended to think like this, because if you have food, right, you don't keep it in the refrigerator. If you keep it lying outside, you get all kinds of living organisms, right? I mean, it stinks, and so they're thinking that it probably is generated from scratch, right? I mean, that there was a spontaneous regeneration theory. So Pasteur was able to put an end to that by shutting off the air supply, right? Over there, like make it in a vacuum, right? And then there's no generation. He was able to put that to rest, So we know now that new life has to come from the only way to produce new life is from existing life, right? There's no other way, right? And cell division is at the heart of it, right? So cell division plays a crucial role in the propagation of all life. The only way to make more cells is by division of those that already exist. A cell reproduces by carrying out a tightly controlled sequence of events in which it duplicates its contents and divides in two. This cycle of duplication and division, which is known as the cell cycle, right? So the cell cycle, the life history of a cell is, divide, right? Then grow, divide, grow, keep on doing that, okay? So this is the essential mechanism by which all living things reproduce. In unicellular organisms such as bacteria or yeast, every time you have a cell division, you get a completely new organism. While in multicellular organisms, many rounds of cell division are required to make a new individual from the single celled egg, right? Like we have, each one of us, we have about 100 trillion cells, okay? So you need that many cell divisions. And this has to be sustained because the cells are replaced all the time. On your skin once every 10 days, okay? In your gut once every 3 days, right? And so on. Now, in multicellular organisms, quite often cell division has to be carried out to sustain the organism in the steady state. So, if the cell division machinery in your body decides to take rest, okay, you're basically dead, okay? So the details of the cell cycle vary from organism to organism and at different stages in an organism's life. Certain characteristics, however, are universal. For example, the cell that is undergoing division will have to replicate its DNA, right? You want the DNA to be faithfully replicated and partitioned between the two daughter cells, right? That's cell division. If there are mistakes, the consequences could be tragic, right? So the cell that is undergoing division must replicate its DNA and pass on identical copies to its two daughter cells. Prokaryotes or bacteria, right? Again, prokaryotes means they don't have a kernel, right? Prokaryon or a nucleus. So prokaryotes or bacteria do not have a nucleus and they reproduce by a fission type of cell division, right? So the DNA replicates, the chromosome in bacteria is usually just a circular chromosome. The DNA over there replicates and one copy moves towards each end of the dividing bacterium, right? And then the cell wall and the plasma membrane in the center of the dividing bacterium, they pinch inwards, resulting in two daughter cells, right? So there is just replication of the circular chromosome, one copy moving to each side, then pinching in the middle, you got 2 cells. All right. Now the cell division in a eukaryotic cell is much more complicated. So with bacteria, there's no cancer or anything like that. It's a unicellular organism, right? So you don't have to worry about cancer. But cell division in a eukaryotic cell is much, much more complicated. That's because most of the genetic information of the cell, namely its nuclear genome, is distributed between multiple chromosomes contained in the nucleus, right? And in addition, the cytoplasm contains many organelles, right? These compartments, many of them, which must be duplicated and apportioned out equally between the 2 daughter cells at cell division. So it's more complicated. Now, there are 3 major questions that have to be answered in the context of cell division. First one is the mechanisms by which the cells will duplicate their contents. The cell contains DNA, RNA, proteins, many different kinds of molecules. Okay, how does it duplicate all this stuff, right? The second question that you have to answer is the mechanisms by which the cell is going to partition the duplicated contents and then split in 2, right? And the third one is that the mechanisms employed by cells to ensure that the different steps involved in cell division take place in the proper sequence, right? There has to be some kind of a timing that controls the entire process, right? Just like in a computer, you need timing, you need a clock, right? There has to be some kind of clock, right? So partial answers to the first question have already been provided in earlier chapters. For example, we discussed DNA replication, right? So how do you duplicate DNA? We know that, okay? You use DNA polymerase and all those different steps, right? Then we also discussed protein synthesis. That's how the cell is going to replicate protein, right? And then there are other organelles also. We have not done the details of that. They are there in any book on molecular biology. In this chapter, we are going to focus on providing an answer to the second question, that is the mechanisms by which the cell is going to partition the duplicated contents and split in 2, right? And the answer to the third question, which is about the timing of the cell, right? How does it time the onset of cell division and so on? That will be taken up in the next chapter, right? Now, the duration of the cell cycle varies greatly from one cell type to another, one type of cell to another. For example, a single cell yeast can divide every 90 to 120 minutes under ideal conditions, while a mammalian cell, liver cell, will divide on an average less than once a year. Okay, so they vary. Okay, some cells divide a lot faster, some never divide. For example, nerve cells do not divide at all. Okay, so in this chapter, we will focus on the sequence of events in a fairly rapidly dividing mammalian cell with a cell cycle time of about 24 hours. Now, the eukaryotic cell cycle is broadly divided into 2 phases. Right? One of them is called M phase, right, which is composed of mitosis. Mitosis is nuclear division. Okay? This is the process by which the nucleus of the cell is split in 2. Right? That is what mitosis is. So M phase is the combination of mitosis, that is nuclear division, and what is called cytokinesis, which is the splitting of the cell in 2. Right? So the nuclear division, and then the cell has to split in 2, right? That's M phase. And then the interphase is the period between one M phase and the next, okay? So every time you're having a nuclear division and cytokinesis, then after that there is some period in between, right? The cell is going to grow and then again divide, okay? Grow, divide, okay? If it's in that cycle. There are cells that have withdrawn from that cycle, like nerve cells never divide. They have to last a lifetime, and that's why if you have nerve injury and all that, you're basically, in pretty bad shape. You don't recover, because those cells are not going to divide. Right? Now, the interphase, that is the period between one M phase and the next, right, is further divided into the remaining 3 phases of the cell cycle as shown in the following figure. And again, you don't have to memorize for this exam, but for the next one, you will have to, okay? You need to know this, that see, you have this M phase is mitosis and cytokinesis. Okay, mitosis is the nuclear division. Cytokinesis is the actual splitting of the cell in 2. All right, that's M phase. So in between one M phase and the next, you are going to have interphase. All of this is interphase. Okay, but in this interphase also you have, different classifications. The DNA replication phase is called the S phase. Okay, S is for synthesis, DNA synthesis phase. All right. So S phase. So, if you ask me, you have to memorize this. You cannot call it some other name. Everybody calls it S phase, right? Then you also have G1 phase and G2 phase. G is for gap, right? G1 phase is the gap between M and S, right? This is the gap between M phase and S phase, right? This is called the G1 phase. And the other one, gap between S phase and M phase is called the G2 phase. So during this G1 and G2 phases, these gap phases, the cell is going to grow, all right, increase, and is going to produce proteins and whatever else it needs, right? Because otherwise, every time it divides, the cell will become smaller, right? Right? It needs to acquire more material, right? So you have M phase, G1 phase, S phase, G2 phase, right? And this is the complete cell cycle, right? Yeah, go ahead.
So it starts from M phase or starts from G2 phase?
Well, M phase is when it divides, actually, okay? M phase is when the nucleus will divide.
Which one first happen? Is G2 phase or M phase?
No, this is a cycle, so it doesn't matter, it can start anywhere, If you ask which one first happened, it's like asking which one came first, the chicken or the egg, I don't know the answer to that, okay? But I'm saying once you're in the cycle, right, the cell has to grow, there's growth here, there's DNA replication, growth, right? There's enough growth, all right? So everything has been duplicated. Then the cell divides during M phase, okay? Again, it'll grow. Then there'll be DNA replication, grow some more, and when it's grown enough, again it'll divide. And that's how the cell division will take place, yeah. Okay, did that answer your question? Yeah, I don't.
No, I mean like G2 phase is to prepare for
Mitosis, yeah.
Yeah, and so I think the G2 is will first happen and then M phase.
Yeah. I mean, the cell has to be big enough, yeah. Yeah. So G2, well, it's like once M has happened, all right, then after that, so now you got two cells, okay? But before these cells can divide again, they need to grow, okay? So there'll be this growth during this, then there'll be DNA replication, some more growth. In fact, if this thing is very well timed, we are not discussing in this chapter. What happens if the cell hasn't grown enough? There are molecular breaks that will stop the cell division, okay? If the DNA didn't replicate, let's say if the DNA got damaged, all right, you don't want to replicate damaged DNA. That has to be repaired. That means you have to slow down, okay? You cannot go and just go and do all these things, okay? There are breaks in the cell. There are molecular breaks that will prevent the progression of the cell cycle, right? It will slow things down so that you can repair and do the work. And in fact, the analogy that is given in the book corresponding to this is like a washing machine, all right? The clothes have to go through the rinse and the wash and all that. So you don't just go and, if one process is taking too long, you don't just go and start the next one. You cannot do it out of sequence. So it's the same thing here. Yeah.
For the S phase finish, then it's like you have every nuclear cell for chromosomes.
Right. The DNA has replicated, yes, yeah.
So it has to, okay.
No, but that's not the only thing, okay? All these other organelles, everything has to be replicated, okay? The DNA is only part of the story. Yeah, you do need the DNA to be replicated, and you have to have enough DNA so that you split it apart. But all the other things also have to be replicated.
Chromosomes are also formed within this S phase.
Oh, yeah. Because chromosome is just a packaging of the DNA. Okay, yeah. So S phase is when the DNA is going to get replicated. Okay. And in fact, you need to know this terminology like G1, G2S and all that because, let's say you zap some cells with radiation, okay, huge dose of radiation, right? Then you damage the DNA, right? So what should happen? You don't want the cell to propagate with damaged DNA. So there should be DNA repair, right? So because of that, it will arrest in the G1 phase, okay? It will not replicate the DNA with the damaged DNA. So they call this G1 arrest, okay? So if you don't know these phases, if you see in a paper they talk about G1 arrest, okay? You'll be wondering, you'll think some cop arrested somebody, It's really this G1 phase that they're talking about, okay? Growth arrest. But we'll, discuss some of these things a little bit later, okay? Yeah, go ahead. DNA what? Oh, you mean you're talking about homologous recombination and all? It can happen. You hope that it doesn't, okay? Because if it does, then there can be mistakes, okay? I mean, like if there are mistakes, then it's a problem. Especially, see, this type of cell division is still okay because it's for the body cells of the body. This is for the somatic cells, okay? But if in the reproductive cells, if there are mistakes, that's a problem, okay? Like if you've seen people with Down syndrome, they got an extra copy of chromosome 21, okay, because of some mistakes in this thing. So yeah, there could be shuffling and all that, but usually, the cell tries to make sure that the DNA replication is as faithful as possible, So hopefully if there are mistakes, if there is shuffling and all that, as something abnormal, right? That's not the normal thing. So during interphase, you have this S phase, which S is for synthesis. DNA replication will take place during S phase. The S phase is flanked by two phases where the cell continues to grow. The G1 phase, G is for gap, is the interval between the completion of the M phase and the beginning of the S phase. And the G2 phase is the interval between the end of the S phase and the beginning of the M phase. Right? I show that to you on the picture. Now there are particular times in the G1 and G2 phases when the cell will make a decision whether to proceed to the next phase or pause to allow more time to prepare, right? Like if you don't have enough food, okay, there's no point for the cell to create more, descendants, right? So things like that, those factors could weigh in the cell's decision whether to divide or not. So these times are called the G1 and G2 checkpoints respectively. Now during all of interphase, the cell continues to transcribe genes, right, to produce the necessary, messenger RNA, synthesize proteins, and it will grow in mass, right? Together, G1 and G2 phases will provide additional time for the cell to grow and duplicate its cytoplasmic organelles, right? Everything has to be duplicated because every time the cell divides, you don't want the cell to become progressively smaller and smaller, right? There are cell divisions where that happens, and I think that's there on the next slide. Now, if you didn't have the G1 and G2 phases, you just had S, DNA replication, then cell division, okay, S, M, S, M, right? Then the cell would get progressively smaller at each cell division, right? And this is in fact what happens to a fertilized egg during the first few cell divisions of its life, referred to as cleavage cell divisions, where it's just splitting, right? The growth is not there. Now the first readily visible sign under a microscope that a cell is about to enter the M phase, right, that you're going to have nuclear division, is the progressive condensation of its chromosomes, right? This is how cell division was first observed in the late 1800s, okay, under a microscope, you can see that. So this condensation will make the chromosomes less likely to get entangled and therefore physically easier to separate during mitosis. And the separation of the duplicated condensed chromosomes is carried out by a transient structure which we will discuss in detail, which is called the mitotic spindle, right? And for animal cells, the actual division of the cell, the cytokinesis, right? Because the nuclear division is mitosis. Okay, after that you need the splitting of the cell. The actual division of the cell is carried out by a contractile ring, right? So it's like the nucleus has doubled. Okay, it's split up in two, and then in the middle there will be a ring that will just pinch, and the cell will divide in two, right? So for animal cells, the actual division of the cell is carried out by what is called a contractile ring, made up of two kinds of protein filaments, actin and myosin. Now, like the mitotic spindle, the contractile ring is also a transient structure which assembles at the appropriate time, only when it's needed. It's not there all the time. Now, in plant cells, the cytoplasmic division has to be carried out by a different mechanism because each cell is enclosed within a hard cell wall. So this is a difference between plant and animal cell division. Now, small organelles in the cell, such as mitochondria and chloroplasts. Okay, so mitochondria, what is the function of mitochondria in cells? Energy. Okay, yeah, good. You guys have been studying for the exam. Okay, so chloroplasts is, yeah, in plants. Yeah, good. So are usually present in large numbers in each cell and will be safely inherited by the daughter cells if on the average their numbers simply double once every cell cycle, right? For other larger organelles, such as the Golgi apparatus and the endoplasmic reticulum, right, these will disintegrate into small fragments during mitosis, which increases the chance. So they have become many small pieces, then they'll be apportioned, okay, approximately equally between the two, and then they'll reassemble, right? So subsequently, the organelles are reconstructed from the inherited fragments present in each daughter cell, right? Now, we next take a detailed look at mitosis, which is the type of nuclear division that most eukaryotic cells undergo. Now, before mitosis begins, each chromosome has been replicated, right? The DNA has replicated, right? And consists of two identical chromatids, or which are called sister chromatids, which are joined together along their length by interactions between proteins on the surface of the two chromatids. A typical pair of sister chromatids is shown in the figure, right? I think it's in the next figure, okay? So once the chromosomes have replicated, right? So these are the two replicated chromosomes. They are attached here, right? They are called sister chromatids, right? And there is a location here that is the centromere region. There is a telomere, centromere, right? replication origin, right? Centromere is where some protein called kinetochore will come and assemble, and that's how the things that pull the two chromosomes apart, right? That is called the mitotic spindle is going to come and attach on the kinetochores. Now, although mitosis proceeds as a continuous sequence of events, it is traditionally divided into 5 stages. Okay, everything appears to happen at the same time, but it's studied in 5 stages. The first one is prophase. The next one is prometaphase. Again, for the next exam, all of these names have to be memorized. Prometaphase, metaphase, anaphase, and telophase. All right, and we would discuss these phases one by one. Okay. First, a brief description, then I'll stop for today. All right, and then after the test on Monday, we'll talk about them in detail. So during prophase, what happens is that the replicated chromosomes will condense, and the mitotic spindle will begin to assemble outside the nucleus. The next phase is prometaphase. During prometaphase, the nuclear envelope will disintegrate, and these spindle microtubules can come in contact with the duplicated chromosomes. Now, during metaphase, the mitotic spindle will gather all of the chromosomes to the center equator of the spindle, right? So you have these 2 spindles like this, and then the chromosomes will line along the equator, right? Now, during anaphase, the 2 sister chromatids in each replicated chromosome synchronously split apart, and the spindle draws them to opposite poles of the cell. And during telophase, a nuclear envelope reassembles around each of the 2 sets of separated chromosomes to form 2 nuclei. And in 3 of the stages described above, we have referred to the mitotic spindle. A natural question that comes up is, how is the mitotic spindle formed and what exactly is its role? So I'm going to start with this slide next time. Okay? I'll again go through this because all this stuff we are going to elaborate on in the subsequent slides. Okay? Each of these stages. So I will see you guys on Friday. Right? Come well prepared for the test and go do a good job, and then everybody is happy. Grading for me will become like a piece of cake. So.